But life, of course, consists of final partings as well as first meetings. And as much as I and my family miss him, I know he would want us to enjoy Christmas. We felt his presence as we, like millions around the world, readied ourselves for Christmas.
They tested Queen Elizabeth I's DNA, and what they found left scientists speechless. For over 400 years, the world believed they knew everything about England's most powerful queen. But when researchers finally extracted genetic material from her personal belongings, they uncovered a horrifying secret that had been literally embedded in her cells. The truth about what was happening inside her body while she ruled an empire is absolutely shocking.
We see our own children and their families embrace the roles, traditions, and values that mean so much to us, as these are passed from one generation to the next, sometimes being updated for changing times.
What scientists discovered hiding in her DNA changes everything we thought we knew about the Virgin Queen, and you won't believe what it reveals about her final years. Elizabeth I ruled England from 1558 to 1603, a period that historians often call the Elizabethan era. Her reign saw the defeat of the Spanish Armada, the flourishing of English drama with writers like William Shakespeare, and the establishment of England as a major world power. Yet despite her prominence in history, books Elizabeth herself has remained something of an enigma. We know her through painted portraits, through letters she wrote and received, and through accounts written by people who knew her. But until now, we have never had access to her biological information, the kind of data that can tell us about her health, her ancestry, and the physical realities of her daily life. The announcement came after years of quiet work by a team of geneticists and historians working together on an ambitious project. When the news broke, it caught many people by surprise. The idea that we could learn something new about a queen who died more than 400 years ago seemed almost like science fiction. Yet the research team had developed new techniques that made it possible to extract tiny amounts of genetic material from objects that Elizabeth handled and wore during her life. The project faced a significant challenge from the very beginning. Direct sampling from royal remains has always been considered inappropriate for a variety of reasons. There are ethical concerns about disturbing the dead, cultural sensitivities around treating royal bodies as research subjects, and political complications that arise when dealing with historical figures who still hold symbolic importance. These barriers had kept scientists away from direct examination of royal remains for generations. The research team needed to find another way forward. The breakthrough came when they realized that modern DNA recovery techniques had advanced far enough to work with indirect sources. Instead of trying to access Elizabeth's tomb, they turned their attention to items she had used regularly during her life. Museums and archives across England have preserved many objects from the Tudor period, and some of these items had belonged to Elizabeth herself. The team focused on things like gloves, lace collars, embroidered gowns, and even cosmetic tools that had been kept in carefully controlled conditions for centuries. These objects turned out to be treasure troves of biological information. When a person wears clothing or handles objects, they leave behind microscopic traces of themselves. Sweat, skin oils, and tiny fragments of skin cells all transfer to fabric and other materials. Some of Elizabeth's items even contained residue from lip products and herbal powders that she had applied to her skin. All of these substances potentially contained her DNA, preserved by accident across the centuries. Extracting usable genetic material from such old sources required the development of entirely new protocols. The research team drew on techniques from paleogenomics, which deals with ancient DNA, and proteomics, which studies proteins. They had to work with extreme care, knowing that many of their attempts would fail. Ancient DNA degrades over time, breaking down into smaller and smaller fragments. The material they were working with was not just old, but also contaminated with DNA from everyone who had handled these objects over the years, from servants in the Tudor court to museum curators in modern times. The process demanded extraordinary patience. For every sample they processed, the team had to account for contamination, verify that the genetic material they found actually dated to the 16th century, and confirm that it could plausibly have come from Elizabeth rather than from someone else who had touched the same objects. They used control samples and statistical analysis to separate genuine signals from background noise. The work took years, with many false starts and dead ends along the way. Then, gradually, the pieces began to fall into place. The first sequences started to assemble into coherent strands of genetic information. When this happened, the researchers realized they had accomplished something that had seemed impossible just a few years earlier. They had recovered analyzable DNA from one of history's most studied figures, a person who had been dead for more than 4 centuries. The implications were staggering. The scientific community responded with a mixture of excitement and cautious skepticism. Some researchers wanted to see the full methodology before accepting the findings. Others immediately began thinking about what questions could now be answered with this new source of information. Historians who had spent their careers studying Elizabeth through documents and portraits suddenly had access to a completely different kind of evidence. Biologists saw an opportunity to understand how genetic factors might have influenced the life of someone whose decisions had shaped an entire era. For the general public, the announcement raised fascinating questions. What could DNA tell us about a historical figure that we did not already know? Could genetic information change our understanding of Elizabeth's personality, her choices, or her legacy? And perhaps most intriguingly, what secrets might be hidden in her genome that portraits and letters could never reveal? The research team had succeeded in crossing a threshold that many had thought would remain forever closed. They had found a way to bring modern science to bear on historical questions in a completely new way. The items that Elizabeth had worn and used, preserved by chance in museum collections, had become windows into her biology. Through these windows, scientists could now glimpse aspects of her life that no historian had been able to access before. As the team began to analyze the genetic sequences they had recovered, they knew they were looking at something unprecedented. This was not just DNA from any person, but from someone whose life had been documented in extraordinary detail through other means. They could compare what the genome told them with what historians already knew from written records. They could test hypotheses that scholars had debated for generations, and they could potentially discover entirely new information that no one had even thought to look for. The project represented a new frontier in historical research, one where laboratory science and archival scholarship met and merged. It showed that the past still has secrets to yield, and that new technologies can unlock information that seemed permanently lost to time. What the scientists found in Elizabeth's DNA would prove to be even more surprising than anyone had anticipated. The first major findings from the genetic analysis came from examining Elizabeth's mitochondrial DNA, the genetic material that passes down through the maternal line from mother to child. Historians had already mapped out Elizabeth's family tree through her mother, Anne Boleyn, with considerable detail. The Tudor genealogies were well documented, tracing lineages through the English aristocracy and connecting various noble families across generations. Everyone expected the DNA to confirm what the historical records already showed. Instead, the results pointed to something far more interesting. The mitochondrial sequences revealed ancestry that was more layered and complex than anyone had anticipated. While the DNA certainly showed markers consistent with English aristocratic lineage, it also displayed genetic signatures that researchers more commonly associate with populations from the Mediterranean region. These markers suggested that somewhere in Elizabeth's maternal ancestry, possibly generations before Anne Boleyn, there had been mixing between families from different parts of Europe. This was not entirely shocking to historians who study medieval and early modern Europe, but it added concrete biological evidence to what had previously been only speculation and rumour. Court gossip from Elizabeth's own time had occasionally hinted at exotic heritage in various noble families, but such rumours were often dismissed as mere scandal or political ammunition. The genetic data suggested that there might have been truth behind some of these whispers. More importantly, it illustrated something that historians have increasingly come to understand about European noble families. They were not as isolated or homogeneous as their carefully curated family trees might suggest. Marriages, travel, and various other connections created genetic diversity, even within families that seemed to marry only among themselves. These findings about ancestry did not change Elizabeth's identity as a Tudor or alter the historical record of her parentage. What they did was add depth to our understanding of how medieval and early modern European populations were connected. Noble families exchanged not just political alliances, but genetic material across regions that we might now think of as quite distant from one another. The Mediterranean markers in Elizabeth's maternal line spoke to centuries of movement and connection that had shaped the genetic landscape of European aristocracy. Beyond ancestry, the DNA revealed information about Elizabeth's physical characteristics and health predispositions that gave new context to things historians already knew from written records. For generations, scholars had puzzled over various aspects of Elizabeth's health and behaviour based on letters, diaries kept by court members, and official records. Now, genetic variants provided possible biological explanations for patterns that had been noticed but never fully understood. One particularly significant finding involved a genetic variant associated with severe migraines. Historical accounts from people who lived at Elizabeth's court had described episodes when the Queen would suddenly withdraw from public view, cancelling appearances and retreating to her private chambers. These incidents had been documented but never fully explained. Some historians had attributed them to political strategy, others to temperament or mood. The genetic evidence suggested a simpler explanation. Elizabeth may have suffered from debilitating headaches that forced her to seek isolation and quiet until they passed. This discovery transformed how researchers read certain historical documents. Passages that had seemed mysterious or had been interpreted as evidence of political maneuvering now appeared in a different light. They became records of a woman managing a painful medical condition while trying to maintain the demanding schedule of a ruling monarch. The DNA did not prove definitively that Elizabeth had migraines, but it showed she carried genetic factors that would have made them significantly more likely. Another variant pointed to heightened susceptibility to autoimmune conditions. This finding resonated with historical records describing chronic skin problems that Elizabeth reportedly struggled with throughout her life. Various accounts mention her use of heavy cosmetics, which historians had generally interpreted as fashion choices or attempts to maintain a youthful appearance as she aged. The genetic data suggested an additional possibility, that she might have been covering up skin issues caused or worsened by autoimmune responses. Again, the DNA did not prove she had specific autoimmune diseases, but it indicated biological vulnerabilities that could explain symptoms documented in historical sources. These discoveries about health predispositions added a layer of realism to the study of Elizabeth's life. Behaviours and choices that historians had analysed primarily through political or psychological lenses could now be understood partly as responses to biological realities. She was not just a political actor making strategic choices, but also a person managing physical challenges that affected how she could present herself and carry out her duties. The analysis of cosmetic residues provided some of the most striking insights. Elizabeth became famous during her lifetime for her distinctive appearance, particularly in her later years. The white-painted face, vividly coloured lips, and elaborate wigs she wore were not just fashion statements, but carefully constructed elements of her public image. In an era when a monarch's appearance carried symbolic weight, Elizabeth had turned her physical presentation into a tool of political power. She crafted an image of ageless, unchanging authority that helped stabilize her rule during turbulent times. Researchers analyzing residual compounds from her cosmetics found evidence of the chemical interactions between makeup and skin at the molecular level. They detected lead-based substances that were commonly used in face paints during the 16th century. Medical historians already knew that lead-based cosmetics were dangerous, but the new analysis revealed how these substances had interacted with Elizabeth's particular genetic profile in ways that likely caused significant physiological stress. The protein markers recovered from cosmetic residues showed signs of inflammation at levels that modern medicine would consider worrisome. These markers suggested that the very cosmetics Elizabeth used to project strength and beauty were simultaneously damaging her health. The interaction between the toxic compounds in her makeup and her genetic vulnerabilities created a situation where maintaining her iconic appearance came at a serious biological cost. Every time she applied the white lead paint to her face, she was exposing herself to substances that triggered inflammatory responses in her body. This finding created a poignant irony in the story of Elizabeth's reign. She had cultivated an image designed to demonstrate her fitness to rule and her transcendence of ordinary human frailty. Yet the very tools she used to create this image were harming her at a fundamental biological level. The DNA and protein evidence revealed a woman whose commitment to her public role led her to endure significant physical suffering, possibly without fully understanding the long-term consequences of her cosmetic practices. The genetic analysis also addressed one of the most debated aspects of Elizabeth's personal life, her decision never to marry and her lack of children. Historians have offered many explanations for these choices over the years. Some have emphasized political strategy, arguing that marriage would have compromised her power or created dangerous factional conflicts. Others have pointed to personal factors, including possible trauma from seeing what happened to her mother and other women at her father's court. Some scholars have quietly suggested that medical issues might have played a role, but without evidence, such theories remain speculative. The DNA provided new information relevant to this old debate. Researchers identified genetic variants that indicated a high probability of congenital complications that could have made pregnancy dangerous or even impossible for Elizabeth. These were not minor factors, but significant biological considerations that might have created serious risks had she attempted to bear children. The findings did not prove that Elizabeth knew about these risks or that they consciously influenced her decisions. Medical understanding in the 16th century was very different from today, and she would not have had access to the kind of diagnostic information that modern genetics provides. What the genetic evidence did was add another dimension to discussions about Elizabeth's choices regarding marriage and motherhood. It suggested that alongside all the political calculations and personal considerations that historians have long discussed, there may have been biological realities that shaped what options were actually available to her. The pressure on Elizabeth to marry and produce an heir was enormous throughout her reign. Knowing that she may have carried genetic factors that made pregnancy exceptionally risky adds a tragic undertone to a story that has often been told purely in political terms. The research team discovered that the genetic and protein data they had recovered could tell them things that went far beyond simple questions of ancestry and health conditions. As they continued their analysis, they began to find markers that spoke to Elizabeth's lived experience in ways that felt almost intimate, revealing aspects of her daily reality that no portrait or letter could capture. One of the more unexpected areas of discovery involves stress markers embedded in keratin proteins. Keratin is the structural protein that makes up hair and nails, and the researchers had managed to extract fragments that they believed came from wigs and hair accessories that Elizabeth had worn. These protein fragments contained biochemical signatures that can form in response to prolonged physiological stress. The patterns they found indicated cycles of intense stress, periods of inadequate nutrition, and disrupted sleep patterns that had left their mark at a molecular level. These findings aligned with what historical records had long documented about the immense pressures Elizabeth faced as queen. She ruled during a time of religious conflict, international tension, and constant threats, both foreign and domestic. Court documents and letters from her advisors frequently mentioned the weight of responsibility she carried and the relentless demands on her time and attention. But the scientific evidence showed that this stress was not just an external pressure or an abstract burden. It had manifested in her body at a fundamental biochemical level, creating changes in her endocrine system that would have affected how she felt and functioned day to day. The stress markers suggested periods of what modern medicine would recognize as significant endocrine disruption. This is the kind of biological pattern seen in people who experience sustained high-pressure situations without adequate relief or recovery time. The molecular evidence painted a picture of someone whose body was constantly responding to threat and demand, maintaining alertness and readiness even when it came at a physiological cost. These were not occasional spikes of stress, but sustained patterns that persisted over extended periods. What made these findings particularly meaningful was how they connected with historical accounts of Elizabeth's leadership style and her legendary capacity for managing crises. She was known for her ability to remain composed under pressure, to think strategically in moments of danger, and to project confidence even when circumstances were dire. The biological evidence suggested that behind this outward composure was a body working overtime, managing stress through biochemical mechanisms that took a toll over time. Her strength was real, but it came at a price that played out in her cells and tissues. The analysis also revealed information that connected to Elizabeth's cognitive abilities and mental characteristics. Among the genetic variants the researchers identified were several associated with enhanced verbal memory, improved pattern recognition, and rapid cognitive processing. These kinds of genetic markers do not determine intelligence or guarantee specific abilities, but they do indicate predispositions that can influence how the brain develops and functions. Historical records had long celebrated Elizabeth's intellectual gifts. She was known for her sharp wit, her skill with languages, her ability to compose eloquent speeches on short notice, and her talent for reading political situations accurately. She could reportedly recall conversations and documents with impressive precision, and she had a reputation for making quick decisions even in complex situations. These abilities had been attributed to her education, which was indeed exceptional for a woman of her era, and to her natural talents. The genetic findings did not diminish the role of education or personal effort in developing these skills. What they suggested was that Elizabeth might have started with biological predispositions that made certain kinds of learning easier for her. The interplay between her genetic inheritance and the rigorous education she received may have created the intellectual powerhouse that impressed and sometimes intimidated her contemporaries. Nature and nurture had worked together, each reinforcing the other. These discoveries sparked interesting discussions among scholars about how we understand historical achievement. Elizabeth's accomplishments as a ruler and political strategist remain just as impressive whether or not she had genetic advantages. If anything, understanding the biological factors that contributed to her abilities made her story richer rather than less meaningful. It showed how various elements, from genes to education to circumstance to individual choice, combined to produce someone capable of guiding a nation through turbulent times. Another intriguing finding involved genetic variants related to heightened sensory perception. These markers suggested that Elizabeth may have experienced certain sensory inputs, particularly sounds and scents, with greater intensity than average. If this was the case, it could explain several anecdotes recorded by people who observed her at court. Multiple sources mentioned her strong aversion to particular perfumes and her discomfort with loud or sudden noises. These had generally been interpreted as personal preferences, or perhaps signs of a temperamental nature. The genetic data offered a different interpretation. Elizabeth might have been genuinely more sensitive to these stimuli at a neurological level, experiencing them as more intense or even overwhelming compared to how others perceived the same sensations. This would not have been a matter of preference or personality, but rather a feature of how her nervous system processed information from the environment. Such heightened sensitivity could have been both an asset and a challenge, making her more attuned to subtle changes in her surroundings, while also making certain environments uncomfortable or difficult to tolerate. The researchers also found genetic markers that pointed to distinctive metabolic patterns. These variants suggested possible irregularities in how Elizabeth's body processed glucose and managed energy levels throughout the day. Historical records contain numerous references to her eating habits, which observers at the time found unusual. She reportedly had a selective diet and ate at irregular times, often consuming very little at formal meals, but eating at odd hours when others were not present. Historians had generally interpreted these eating patterns as eccentricities, perhaps signs of the control she exercised over her environment, or possibly linked to concerns about poisoning that would have been reasonable for a monarch of her era. The genetic evidence suggested an additional possibility that these eating patterns might have been Elizabeth's intuitive response to metabolic needs that made regular meal schedules uncomfortable or impractical for her. If her body processed food and regulated blood sugar differently than average, she might have developed eating habits that worked better for her particular physiology, even if they seemed odd to observers who did not share those metabolic characteristics. Perhaps the most disturbing discovery came from analysing the residues found on clothing and items from Elizabeth's later years. Alongside genetic material, researchers detected compounds that told a troubling story about the cumulative effects of the substances she exposed herself to regularly. The analysis revealed high concentrations of heavy metals, particularly lead and mercury, which were common ingredients in cosmetics of the period. But more alarming was evidence of how these toxic substances had interacted with her body chemistry over time. The biochemical markers indicated what modern toxicologists would recognize as chronic poisoning. This was not a case of someone deliberately trying to harm the Queen, but rather the result of beauty practices that were standard for wealthy women of her era. The white face paint that created her iconic appearance contained lead. Various other cosmetics contained mercury and other substances now known to be dangerous. Elizabeth had been applying these products to her skin regularly for decades, and her body had been absorbing them all that time. The protein markers showed signs of ongoing inflammatory responses and cellular stress consistent with heavy metal exposure. The interaction between these toxic compounds and Elizabeth's particular genetic vulnerabilities appeared to have created a situation where the cumulative damage was even more severe than it might have been for someone with a different genetic profile. Her commitment to maintaining the powerful visual image she had created was slowly poisoning her, and the biological evidence suggested the effects had been significant. This discovery transformed understanding of Elizabeth's final years. She had continued to present herself in public with the same carefully constructed appearance even as she aged and her health declined. The DNA and protein analysis revealed that this dedication to her image had come at an enormous physical cost. The very cosmetics that helped her project ageless authority were undermining her health at a cellular level, creating a tragic contradiction at the heart of her public performance. The microbiome traces found in the materials added yet another layer of information. Though fragmentary and incomplete, these microbial signatures provided clues about the environments Elizabeth inhabited and the routines that structured her daily life. Different bacteria and other microorganisms leave traces that can indicate what kinds of spaces a person spent time in, what they ate, and how they interacted with their surroundings. For historians trying to reconstruct the actual lived experience of Tudor court life, these microbial hints offered valuable, supplementary evidence to combine with written records and architectural information. When the research team first embarked on their project, their goals had been relatively modest. They wanted to explore whether it was technically possible to extract human DNA from historical artifacts with enough clarity and reliability to produce meaningful scientific results. The work was experimental, pushing the boundaries of what paleogenomics could accomplish. They expected that if they succeeded at all, they might be able to confirm some basic genealogical information, or perhaps identify one or two genetic variants of historical interest. What they actually discovered went far beyond anything they had imagined when they started. The genetic and protein data they recovered told a story about Elizabeth that was both more complex and more human than the carefully polished image she had projected during her lifetime. The analysis revealed a woman whose body carried the accumulated marks of her era, her responsibilities, and her personal struggles. It challenged the idealized portrait that had been passed down through centuries of historical writing and popular imagination. Instead of a distant figure defined primarily by political achievements and royal authority, the DNA presented Elizabeth as someone navigating real biological limitations and vulnerabilities, while simultaneously shouldering extraordinary responsibilities. These findings opened up important discussions about how biology and history intersect. Historians have always known that historical figures were real people with physical bodies, but they have typically had to rely on written descriptions, artistic representations, and occasional medical records to understand the physical dimensions of their subjects' lives. The ability to examine actual genetic and biochemical evidence from someone who lived hundreds of years ago created entirely new possibilities for historical research. It demonstrated that the biological reality of historical figures can be studied directly rather than just inferred from secondary sources. The scientific evidence about Elizabeth's health challenges, stress markers, and the effects of her cosmetic practices did not diminish her legacy or reduce her accomplishments. If anything, these discoveries made her achievements seem even more remarkable. Understanding that she may have dealt with chronic pain from migraines, managed autoimmune symptoms, endured the physiological effects of lead poisoning from her makeup, and coped with sustained biochemical stress while simultaneously running a kingdom-added depth to appreciation of her strength and resilience. She had guided England through religious conflicts, survived multiple assassination plots, overseen the defeat of the Spanish Armada, and presided over a cultural flowering, all while dealing with physical burdens that would have challenged anyone. The project also raised fascinating questions about how genetic factors might have influenced the personalities, decisions, and leadership styles of historical rulers more broadly. If Elizabeth carried genetic variants that affected her cognitive processing, sensory perception, stress responses, and metabolic function, then it seems reasonable to suppose that other historical figures likewise had biological characteristics that shaped their experiences and capabilities. This does not reduce historical events to biological determinism, but it does suggest that the human bodies behind major historical developments may have played roles in shaping outcomes in ways that traditional historical analysis has overlooked. The genetic evidence about Elizabeth became a focal point for scholars interested in developing this new interdisciplinary field. Her case was particularly valuable because so much documentation already existed about her life, creating opportunities to compare biological findings with written records. Researchers could look at what the DNA suggested about her cognitive strengths and then examine historical accounts of her decision-making. They could see genetic markers for stress responses and compare them with documented periods of political crisis. This interplay between biological and historical evidence created a richer, more dimensional understanding than either type of evidence could provide alone. Beyond academic circles, the discoveries sparked broader cultural conversations about beauty standards, leadership expectations, and the hidden costs of public performance. The revelation that Elizabeth had likely suffered significant health damage from the very cosmetics she used to project strength and authority resonated with contemporary discussions about the pressures faced by public figures, particularly women, to maintain certain appearances. Her story illustrated how societies impose visual expectations on leaders and how meeting those expectations can exact physical tolls that remain invisible to the public. These parallels to modern experience made Elizabeth's story feel unexpectedly relevant despite the centuries separating her era from ours. The biological evidence showed that the pressure to project an idealized public image while concealing vulnerability and managing private struggles is not a new phenomenon. Elizabeth had navigated these challenges in the 16th century using the tools available to her, including cosmetics that poisoned her and a public persona that required constant maintenance. The molecular evidence of what this cost her, physically, invited reflection on similar dynamics in contemporary culture. The findings also stimulated thinking about the nature of historical memory and how we construct our understanding of the past. For hundreds of years, Elizabeth had existed in public consciousness as an almost mythic figure, defined primarily by her political success, her famous speeches, and her iconic visual image. She had been the subject of countless biographies, plays, films, and scholarly studies, all attempting to understand and portray her character and significance. The genomic analysis introduced an unexpected intimacy into this long relationship between Elizabeth and those who study her. The DNA revealed details about her biological struggles and internal experiences that no contemporary observer had recorded and no historian could have guessed. It provided information about pain she likely felt, stress her body managed, and vulnerabilities she probably worked hard to conceal. This created a tension between two different portraits of Elizabeth, the monumental figure celebrated in history books and the individual revealed through her genetic code. Reconciling these two images required acknowledging that she was simultaneously both, a person of extraordinary capability and achievement who was also subject to the same kinds of physical limitations and suffering that affect all humans. Despite the remarkable illumination these findings provided, many questions remained open. The genetic data answered some historical puzzles, but raised new ones in the process. It provided evidence for certain interpretations while complicating others. This is typical of good scientific research, which tends to open new avenues of inquiry rather than simply closing existing debates. The discoveries about Elizabeth's DNA invited scholars to think more carefully about how much of history rests not only on documented decisions and public events, but also on the unseen workings of human biology. Elizabeth's genome became a testament to the intricate relationship between biology and the course of a life. It showed how genetic inheritance, environmental exposures, and individual choices interact to shape human experience. The variants in her DNA had given her certain predispositions and vulnerabilities, but what she did with those biological starting points was shaped by her education, her circumstances, her ambitions, and her choices. The DNA did not determine her destiny, but it was part of the complex web of factors that made her who she was. What scientists uncovered was shocking, not because it revealed weakness or failure, but because it brought Elizabeth's humanity into sharper focus while simultaneously reinforcing respect for her accomplishments. Her genome held traces of pain, endurance, complexity, and resilience that spoke across the centuries. It embodied a fundamental paradox. She had projected invulnerability and timeless authority while simultaneously enduring profound physical strain and biochemical stress. The genetic evidence allowed the world to see not just the public monarch, but the private person who inhabited that role. The project marked a significant turning point in how historical figures might be studied and understood going forward. It demonstrated that the past still contains hidden information waiting to be discovered through the application of modern scientific techniques. Elizabeth's DNA stood as a bridge connecting different eras, a biological text that offered new insights into someone who had been intensively studied for generations. The implications extended outward to historians, scientists, and general audiences, prompting reflection on how deeply intertwined human biology and historical events truly are. Through the lens of genetics, Elizabeth's life appeared both more extraordinary and more relatable than it had before. Her experiences, once filtered primarily through formal portraits and carefully composed letters, became grounded in biological reality. The findings revealed that beneath all the grandeur of her reign was a story about a body navigating challenges of illness, chronic stress, and environmental toxins. Her triumphs were therefore not only political, but deeply personal, achievements of endurance and will that resonated in her very cells. The DNA had given voice to aspects of her story that had been silent for more than 400 years, enriching our understanding of one of history's most studied and fascinating figures.